Okay, happy Monday evening. I appreciate everyone who joined me today. We're going to start in just a few moments, but before we start, what I'd like us to do is to go through a couple of practical things. But I'm going to give a couple of minutes for the last people to join us. So if you would not mind, I would appreciate you posting your name to the chat so I can again take the attendance from there as with the previous times. So thank you everyone who posted their name on the chat for the attendance. I appreciate it. It's a great help. Now I should be able to get hold of the material. So what I wanted to do before we get any further was to just kind of have a look where we're standing with the schedule for this course. Where are we? What's still to do? And what the remaining weeks look like. So I believe that we're now on the week 13. So we have a session today. However, I have not scheduled a class for the coming Wednesday, which is the 17th, simply because we're going to cover the chapter 14 today. And then next week, we will cover the chapter 15. On these two chapters, I've kind of bundled them together, so you will end up noticing that the learning module for both is shared, and you have a quiz that covers the material from both of these chapters. So you are, of course, welcome to make a start of it already this week, or you can wait until next week and complete the entire quiz with all of the questions back then. So I have to just change those chapter quizzes. I think that what I have there, it's all packed up in one single quiz. Then next week, we will also have a Thanksgiving. So remember, not so much for this class, but if you're taking any other classes, that on Thursday, the college is closed, and college will remain closed for the remaining of the week. Then when we return from the Thanksgiving break, we will have one more session. And during that session, we will revise the material for the exam 4, which is the final one of these 4 exams that you've completed or will complete for this course. The finals week I have reserved for course feedback and a couple of extra credit activities that you'll also get a chance to do just in case you want to boost up your grade. But really what I'm wanting to do today is to simply go through the material for the chapter 14, where we will be looking at the DNA, and then the last chapter, chapter 15, where we look at the journey from the genes to the proteins, that I'm saving for the next week, a week from now. So I hope that that makes sense. I know I have a couple of messages that I have received that I haven't had a chance to get back yet to. I think that it's just as busy time for students as it is for us instructors. So if you have something urgent that you haven't received a response and you really, really, really need that response, you can just stick around at the end of this class and we will tackle that then. Does anyone have any questions at this point about the chapter as it relates to, or about the material as it relates to the remaining of the semester? We don't have one final exam that would review everything. You simply complete these 4 exams. I've decided or designed this course so that there would be low stakes, so you don't have too many chapters to cover all at once. And for the chapter 4, we will work through the study guide in case I get a chance to put one together for you. We'll work through that on the week after the Thanksgiving. Well, I don't see any questions coming in. So what I'm going to do at this point, we will simply move on and we'll talk about the chapter 14 as it relates to the DNA. So we've already talked a little bit about DNA, and this will be a chance for us to kind of explore that a little bit more, explore some of the key things that you should walk away knowing about the DNA. So let's see if I get all of the pointers working. So what we saw last time, and this doesn't just limit to Gregor Mendel, this relates to all other early researchers who worked with the genetic materials. You remember that Gregor Mendel, rather than using terms genetic material or DNA, he talked about these minute particles. That's simply because at that time we yet didn't have a name for what he was seeing, but he was observing some outcomes of these minute particles. He just didn't have the terminology yet, but yet he was doing quite a remarkable job of, if I say, of really following what the genetic material does, what it is, and all sorts of things that can happen with it. So the early researchers, including Mendel, but also others, they saw three important things as it relates to the genetic material. First of all, one of the things that they noted was that all of this genetic material had to be able to store information. And this information was used to control the development and also the activities of cells. And through cells, then tissues and organs, organ systems, and organisms as a whole. But so far, if we reflect back to what we have discussed on this chapter, we started with the cells. And what we're really seeing, that we're kind of returning kind of a circle, closed circle moment to those cells. And we're seeing that that information that was stored as genetic material, that was what was controlling the development and activities of these cells. Another thing that these early researchers had noted was that the genetic material had to be able to be replicated. And through that replication process, we're able to pass it to the next generations. So again, you might remember Gregor Mendel's work with the garden peas, where he was able to be quite certain of what the genetic material encoded in one plant was because of the characteristics that he observed from that plant. And remember, he used true breeding plants, so he had bred these plants multiple generations, so he could be really certain that what the phenotype was a good reflection also of the genotype of that plant. But he was also able to observe that as the plant reproduced, the genetic material was replicated and passed to the next generation. The final thing that I wanted to discuss here relates to the concept of mutations. And mutations, when we talk about DNA, when we talk about genetics, tend to have a little bit of a bad ring amongst most people. Most people think of the negative sides of mutations, and there are some for sure. But we also have to remember that really most of the mutations will not have any effect on the offspring. They either are not functional or they're coded to that junk part of the DNA. It will not change what happens. Every now and then there is a mutation that will alter that organism. And sometimes these mutations, as we discussed, might be negative. But this is not the case always. Sometimes we might be introducing something new to the organism, And this new characteristic that we're seeing might actually be beneficial for the offspring. So this is how the concept of evolution works. So in evolution, as we think of, the individuals that have the best possible characteristics are able to get most access to resources. And because they have most access to resources, they of course then appear as very healthy, very successful, and they are attractive mates to other individuals. And since they are attractive mates, they are more likely to produce more offspring. And as a result, those traits that made them more successful and more attractive are passed onwards to the next generation, and so on and so on. So really, it is all about hitting that genetic jackpot that I spoke about a few chapters ago. And sometimes it might not be that we hit that jackpot simply by getting the ideal mixture of genetic material from our parents. Sometimes these ideal characteristics might arise also as a result of random mutations that are beneficial to us. So I guess that's a kind of a little bit of a reminder of us how this all ties into the bigger picture. And if you're interested about how evolution works, survey of life in general, and so on, we do offer the BIO182, where you'll actually get to dive into that much more. This BIO181 is the prerequisite for many courses that you'll be probably wanting to take. BIO182 is not needed for quite as many courses, but kind of expands a lot on these themes that we have discovered on this course. So that's kind of a good setting the scene, understanding what the information that we knew already earlier on was. But I want to remind us about a couple of concepts. So first of all, we remember that all cells have a nucleus. And our today's focus will be on the DNA. And we remember that the DNA was, we're going to talk about what it is, how it affects us, how it works, and all those things. But at this point, you should be comfortable with the basic cell parts, nucleus of the cell, and realizing that the DNA was encoded within the nucleus. So let's take a little bit of a journey back the memory lane, in a sense that we want to understand how did we actually discover the DNA, and what did those discoveries tell us about DNA. And most textbooks, I assume, that you might have seen before, have talked in quite a bit of a detail about James Watson and Francis Crick. It's not really that long ago, if you think about it. Back in 1950s, Watson and Crick set to do two different things. They set to bring together all of the information that we had at that point about DNA. And remember, at this point, we had not seen DNA. We did not have a visual representation. We just knew that it was something that's there and something that's very important. Another thing that Watson and Crick set to do was to build a model of a DNA. And you can actually see that early model of a DNA on the right-hand side of this screen on the picture. And this is what Watson and Crick are really known for being the first to describe and illustrate the structure of the DNA. And what they end up finding out was that DNA could be described as a double helix. So this double helix model of the DNA introduced by Watson and Crick is really their big contribution to the science. And if we think of what a double helix means, Well, we get back to this slide, which I expect to be fairly familiar to you from the back in the day when we first talked about the biological macromolecules. And if you don't remember that, I'll add a link to the very end of this recording if you go back to watch it. But what double helix means is that we have this kind of a stepladder kind of a structure. So we've seen this stepladder kind of structure where we had the side beams, and between them we had these more horizontal poles. The big thing that Watson and Crick noticed that it wasn't like a stepladder as we know. Instead, it was like as if we would have taken this stepladder and rotated it round itself on its axis. So it's kind of this more of a three-dimensional structure where the stepladder is rotated around itself. And this is what we know as the DNA. And we're going to talk about the DNA and its relation to RNA and this actual molecule structure in just a little bit moment. But before that, I want to go back to this picture. So, like I said, this is what most textbooks teach to you, and this is really probably how we've approached the study of the how we've approached this sharing the story behind the discovery of the DNA structure. But there's a little bit more to the story. It turns out that Watson and Crick they were not actually the first ones to figure out the structure of the DNA. A lot of their work actually relied on a picture that was produced by another scientist. And I've shown the very picture to you here on the left-hand side of the screen. So really, I don't think that it would be unfair to say that Watson and Crick used someone else's work to come up with this really revolutionary idea of this double helix stepladder structure that's wrapped around itself. And this picture that you see here was taken by Rosalind Franklin. She didn't think too much of it when she produced it, but really I think that we need to recognize that she should be credited for producing this picture. And what we're seeing in this picture is the DNA structure. We're now looking at it as if we were looking from top down in relation to many of these diagrams that we've seen. So we can see the side beams, and the side beams are slowly rotating as we're looking down. And matching with those side beams, we're seeing those horizontal bars that are running there. So even though Rosalind Franklin was not really in awe of what she produced, but what she produced is behind of everything that we now know to describe the DNA structure. So I think it's fair that we bring her contribution to the forefront. And one of the things that when we're in face-to-face classes that I emphasize quite heavily to my students is the ethics aspect, and the fact that a lot of the science and names and terms in science is the language of dead white old men. So we're using terms that were named after men who discovered things, but history is written by the people who choose what elements to include to the history. And I think that Rosalind's contribution definitely ought to be credited and ought to be recognized. And there are many other female scientists, scientists of color, and so on, who really haven't gotten the recognition that they probably deserve. So that's the little side deviation from what I was talking about. And it brings us back to this diagram that we saw earlier on. And on this diagram, we were starting to look at the DNA. And we know that DNA was stored within the nucleus of the cell, and the function of DNA was to store the genetic information. Well, RNA is another thing that relates very closely to what we'll be talking about and we'll be having a look of. And I thought I had a slide of it, but maybe it's going to come up a little bit later. So RNA is really the workhorse of the DNA. So whereas our DNA is simply there to store the genetic information, RNA instead is there to make something out of this genetic information. And what RNA makes out of the genetic information is typically proteins. So many proteins really drive processes on a cellular level. So proteins are these workhorses, the output, the final product of the information coded within the DNA. And I promised to talk a little bit about the structure of the DNA. We've seen the double helix structure. We saw the side beams. And those side beams would be made by sugar and phosphate molecules. And on this diagram, you can see the sugar as this yellow molecule, and attached to that, we have that phosphate. These two together form these side beams, like I said, of this stepladder model. What we also have, we have a varying component that kind of tends to change around, and this is known as our base. And these bases are what really the varied components form the horizontal lines. And these bases can be of 4 different types. So we can either have an adenine, guanine, cytosine, or thymine for the DNA. An important thing to notice here is that DNA is a double-stranded. So we have those both 2 sides of the stepladder, and then we have these horizontal lines, these bases. RNA instead has a very similar structure, but RNA is made of only 1 strand. So they have still this 1 backbone, if you wish, sugar phosphate backbone of the RNA molecule. And let me just fix that line. Sometimes my screen gets a little sensitive, so this looks better. But unlike DNA, these bases that we're seeing, they don't need to attach to another end. They can just simply be there on their own. Another big difference with the RNA is that although we still have our adenine, guanine, and cytosine, RNA doesn't include thymine. Instead of thymine, we have uracil as a base for RNA. And one of the things that I really wanted to take a little bit of time to discuss with you, and we're going back to this slide, is that there's always a specific pairing of these bases. Of course, as you saw on this diagram that I just saw, there's going to be 2 bases connecting to each other. So what we find is that adenine always connects with thymine, and our guanine always connects with cytosine. Of course, this can be the other way around, that thymine connects with adenine and cytosine connects with guanine. But they are always matched as these pairs. So there's always going to be in the DNA the same amount of adenine and thymine and guanine and cytosine, because these 2 need to be bound to each other on this structure, double helix structure of the DNA. RNA instead, we can have loose ones. We have our adenine, guanine, cytosine, and uracil. They don't need to be bound to anything, these bases. So at this point, I think that a lot of this has been a review. it has been concepts that should be familiar to you from the material that we've talked about. And what I'd like to do now would be to really move on to look at that relationship between DNA and RNA. And look at this journey from how do we go from DNA to make a protein, something useful as a result of it. And what we'll end up finding is that we need one intermediate step, and that intermediate step is going to be our RNA. So what I was hoping to do next with you is a little bit of a drawing. You can either follow me or take your own notes as well, but I'm going to be drawing on this diagram that we have here together. And some of this material, I'm sure I've shared in the past, but we're going to take it a little bit further and add a little bit more detail. So what we just saw was that DNA was this double helix structure. So we have two strands, and they're wrapped around each other. So these were the sugar phosphate backbone that we saw. That looks better. And between these sugar phosphate backbones, we had our bases that were connected. And we remember that these bases had a particular arrangement, which base attached to which one. And it was our adenine to thymine and cytosine to guanine. One thing that we have discussed so far is that DNA is housed within the nucleus of the cell. So what we'll end up seeing is that DNA never leaves this nucleus of the cell. You remember nucleus had a nuclear envelope, so I've just drawn that. And one way how I sometimes describe the DNA is that it's almost like you would go to view an old book within a library. That book is so precious that we never let it leave the library. You go to the special collections part, you're allowed to study it there, you're allowed to take notes, you're allowed to write down things from it, but you never really get to take that book with you. You never get to check it out from the library if you wish. And on this analog, I'm referring nucleus as the library of the cell. Well, what we can do, though, to make some use of this information that we have stored within the DNA, we can make a copy of it. And this copy is something that we're allowed to take away from the library. So this copy would be our RNAs that we make. So let's make an RNA strand here. And what we saw was that RNA was just a single strand. So unlike DNA where we had two strands, RNA will simply have just one strand. It will have the sugar phosphate backbone, and it will also have the bases. But these bases will not need to be attached to a complementary strand of bases. And the big thing that we saw there was earlier on shown here, that rather than thymine, we now have uracil. So we can add that to our little diagram here. So we have our adenine uracil and adenine uracil our cytosine and guanine. They don't need to be matched as we saw earlier on, so they're just simply bases on that RNA strand. So RNA is almost like a photocopy of that library book, that original book that we had, and that's something that we can find either within the nucleus or also outside the nucleus. floating in the cytoplasm of the cell. And this copy contains all the same information that DNA contained, but now this copy probably isn't going to cover all of the DNA. It only covers the section of the DNA that's going to be useful for us for whatever we want to do, whatever we want to make out of the DNA information. So then what happens is that this RNA strand that we had here, it finds a ribosome. And we talked about ribosomes very, very briefly when we talked about the cell organelles. And ribosomes are actually made of two different parts that we see here. And through these two parts, we have our RNA traveling through. And at each time, 3 bases of an RNA are being scanned by a ribosome. So let's add those there. And I'm just, for the sake of simplicity, gonna add that there. And what a ribosome does is that it matches these 3 bases that we're looking at with a proper match of amino acids. Amino acids are found around in the cytoplasm, and we're now going to be looking for the amino acids. And once we find an amino acid that matches these 3 bases that we're looking at one time, we bring it to the ribosome. And that amino acid gets added to the very end of the ribosome. And this happens time and time again with 3 bases at the time. And each time a new amino acid is added to this chain that's forming as a result of the instructions given by our RNA strand. And remember, the instructions of the RNA are simply a copy of the instructions that were stored within the DNA. And as you have a bunch of amino acids as a chain, eventually what they end up doing, they form a protein. So protein is this collection of amino acids that makes something useful, something functional that we can use then to perform functions within the body. So now we have discussed our journey from the DNA to proteins and how it's something useful that we can use as a result of the instructions from the DNA. I want to give 2 more terms that explain these processes that we've seen. So the first term that we're going to see is term transcription. And what transcription tells us, it tells us about the process when we take the DNA and make a copy of that, and that copy was RNA. So it's really a process that has to take place within the nucleus. But once the process has been completed, now we have that information that was previously stored within the DNA, and we can take it outside of the nucleus as an RNA. And with this information that we take outside, of course, we make proteins. So the process of translation refers to taking the RNA and taking the information from RNA and making proteins out of that. So one way to think about this is if you go back to history and look when you were either in a meeting and people still had secretaries who wrote shorthand and all that, what these secretaries would be doing, they were transcribing what was being discussed in that meeting. So it's simply taking a copy, taking notes of the actual information. They were not able to take the actual information because in many meetings that would have been verbal discussion. How do you capture that? There's really no way of taking the verbal discussion and filing that into a filing cabinet. Instead, what secretaries would do, they would transcribe that information and make a kind of transcript of what was being discussed. And in this analog, the transcript that we see would be the RNA. So similarly, if we go back to our library analog, there was that precious historical book where all the recipes for everything that you can do, every dish that you can cook are stored at. That would be our DNA. We take a section of the DNA, so a single recipe that's going to be useful for us, and we make a copy of that recipe. That process is known as transcription. If you think of the process translation then, well, translation refers to giving meaning to something. So rather than just making a copy, we're really dragging out that information and giving it a meaning, making something useful out of it. And that really well describes what happens to an RNA. So RNA carries information, but the RNA itself is not very useful for the cell to make things happen. We need to translate that information into something that actually is able to make things happen. And this thing that's able to make things happen is going to be our protein. So we convert the information from the RNA into a chain of amino acids, and a chain of amino acids is known as our protein. And to do this process, we recruited our ribosomes, that are structures that we discussed earlier on the course when we discussed about the cellular organisms, cell organelles. And again, I'm going to add a link to an end of this recording in case anyone has forgotten those cell organelles, so you can kind of revisit that and hopefully it will ring some bells. So I think that with that, we have done a good job of covering the key points of how we go from DNA to RNA and from RNA to protein. I also will expect you to know these two terms, the terms transcription and translation. And we just visited those, so I hope that that will make sense. So now that we have discussed the general concept of the journey of DNA to RNA to protein, I think what we need to do, we need to look a little bit more detail of what happens in these two events. So what I have here is first the transcription, and we defined earlier on transcription as taking the DNA and the information stored within the DNA and making RNA out of that. So making a copy of that information. Well, so far, what I've told you has always been that DNA was this double helix structure. So we had two sugar phosphate backbones. But what really happens within transcription is that this double helix structure gets opened. So we almost rip apart or separate apart, like Velcro, these two complementary strands of the bases that we saw. So here we have our bases running, matching with their appropriate pairs. And here we have opened these bases, so they're no longer attached to each other, but they would be, again, the complementary matches. So what we saw, we had that, let me take a pencil. We had that adenine always matched with thymine, and our guanine matched with cytosine. So what happens in transcription? We open the double helix structure, and we actually start to build a copy of what has been opened. So remember, what we're really trying to do with the RNA is to simply get that information copied for us. And on this example, I'm going to mark RNA with a different color. So RNA is going to be a strand that we're building. And this strand is going to have bases that match with the bases of the DNA. So one by one, we're adding the right bases so that they match with what we had on the DNA. And you remember the only big difference was with our RNA, that rather than having thymine, we now had uracil. So we have the all other bases as the same, but the uracil is what we'll end up seeing within the RNA. So now we've made this copy of the DNA, and this copy, our RNA strand, that was allowed to leave the nucleus of the cell. So that's how, even though we have some information that we were not able to let leave nucleus, we can copy that information, the actual useful part of that, and we can take it away from the nucleus as this copy. So I hope that that's a good kind of a summary or good explanation of what happens in transcription. And I want to kind of in a similar manner to discuss the process of translation. So in translation, we use RNA to make a chain of amino acids. And a chain of amino acids became a protein. So again, I'm going to use those same colors that we saw earlier on. Here we have our RNA strand. And this RNA strand had bases. Let me actually move our drawing of the RNA strand a little more centrally, so it's going to be easier. So here we have our RNA strand with the sugar phosphate backbone, which is this darker blue, and then we have our bases. And you remember that we had our ribosome, which was the workhorse that we need to actually make some sense, make something useful out of this RNA strand. And this ribosome looked at 3 bases at one time. And what ribosome did, it will match these 3 bases with an amino acid that's a complementary match to this particular combination of 3 nucleotides at the time. And once it's a right match, it's going to bring that amino acid to where it's working and attach it to the very end of what's being produced. And one by one, we will keep adding amino acids to this chain that's being produced based on the instructions that were coded within the RNA. And once we take all of these individual amino acids and now we put them together, they have made a protein. And you remember that proteins were these useful things. They were something that made sense, carried out functions within the cells. So even though the information coded within the DNA and RNA itself is not able to make things happen, we have to take that information and make something useful, and it is the proteins that are usually the ones that make things happen, if you wish. So that, yes, do we have a question? I think I was just hearing my own echo, so we're just gonna carry on. What I do want to do next, I want to compare a little bit of the DNA and RNA. So DNA could be viewed as this kind of a blueprint, if you wish, the instructions for how to make something, whereas RNA led to this final product, the protein. So RNA itself is not the final product, but it will lead to the final product of this process. What we end up finding is that DNA is much more stable than RNA. We end up finding a lot more diversity within the RNA. One way that I described DNA earlier on, that it was almost like this library book that couldn't be checked out, whereas the RNA was a copy of that. That copy you could take away from the nucleus. We saw that the bases that we see at the DNA and RNA are very similar, with the exception of uracil instead of thymine in the RNA. Where we might find DNA? Well, DNA is only found within the nucleus of the cell, whereas RNA is found in all parts, also in the nucleus, but also at the cytoplasm. And we remember those ribosomes being the workhorses of making the protein out of the information coded within the DNA. And here, I'm just going to jump out. I need to change one single term. So I was preparing these slides right before our class, and what we end up finding is that the DNA that survives or stays within the cell, that doesn't really disappear anywhere, whereas RNA, over time, if it's not being needed anymore, it degenerates. And again, let me put a proper letter there. So if RNA is no longer needed, it just ends up being degenerated and it's no longer going to stick around. So that's been our review of the journey of the information coded within the DNA, how that can be converted into RNA, and how RNA then, thanks to the help of the ribosomes, could be converted one amino acid at a time into a protein, and proteins were the ones that actually made something out of it. What I was hoping to wrap up this session with was to talk a little bit about an application of this everything that we have learned. And application that I want to talk about is something that you might have heard about as Dolly the Sheep. So this is a domesticated sheep. Dolly was the first mammal that was cloned from an adult somatic cell. So somatic cells refer to all other cells except the sex cells. So what we did with the Dolly is that we took a cell from the breast tissue of one sheep. We removed the DNA from that cell, so we basically made it into a blank cell without the nucleus. And we took DNA from another organism or from another sheep, and we put the DNA into that cell. And then we gave a shock, electric shock to that cell. And that started that cell to behave like it would have been fertilized egg cell, dividing again and again and again. And then once we saw that that cell was dividing in a successful manner, we implanted it into a carrier another sheep that was ready to carry the pregnancy of this Dolly the sheep that was simply a cell from the breast tissue, but had the DNA of basically a third individual sheep. So in a way, you could say that Dolly had three mothers. So there was the cell mother who donated the cell that was then cleared of the DNA. Then there was the mother who donated the DNA. And then there was the gestational mother who carried the cell to a full pregnancy. Well, the reason why I like to talk a little bit about the Dolly the Sheep is that Dolly the Sheep was cloned in Scotland. She lived to be about 7 years old. A normal life expectancy of a domesticated sheep is somewhere around 11 to 12 years. So she died a little earlier, but not still super early. I believe that Dolly died out of lung disease. She did have arthritis and a bunch of other issues as well. But some of the friends of mine were involved with the process of cloning Dolly. And the reason why Dolly the sheep was so significant milepost in the use of genetics and in cloning was that this was the first mammal that was cloned from a cell other than a sex cell. And as a side story, I sometimes ask questions about side stories, either in the quizzes or in the exams. As a side story that I wanted to share with you is that you might wonder about the name Dolly. That why was this sheep that was cloned, that was the first mammal that was cloned from an adult somatic cell, why was it called Dolly? Well, like I mentioned, Dolly, the cell that gave rise to Dolly, was actually collected from a mammary gland cell. And when the researchers were thinking of a name for this sheep, they could not think of a more impressive pair than those of Dolly Parton. So that's where the name comes from. A little story behind that. And hopefully that helps you to remember. I sometimes do ask, what does Dolly Parton have to do with Dolly the sheep? So that might be a good answer for that. So I think that we have discussed about this technique. This technique, like I said, to take the cell, transfer nucleus from another unfertilized cell to it, and then trigger with electric shock, and then once it's dividing, implanting it into a surrogate. What we're seeing on that process is a technique known as nuclear transfer. So we're transferring the nucleus from one cell or one organism to another cell or to another organism. We have later on been able to do the same process for other large mammals, such as pigs, deers, horses, and bulls, but Dolly was really the first mammal. You might wonder then, well, why was this a big deal? Why didn't they do this earlier on? Or what I get asked is sometimes also that, well, I have a dog that I want to bring back around. Could we do it from the nucleus? What we need to remember is that producing Dolly wasn't an easy task. What happens is that this often results in abnormal development and abnormal cell division. In fact, it took 277 attempts before the cells that gave rise to Dolly were produced. So a lot of the time that gives failures, and it's not very cost-efficient method of bringing cloning something. If we think of the Dolly the sheep and we think, why then would we be doing this? Isn't this a little bit of a concerning thing? There are ethical concerns. for sure to consider, but also this technique potentially would allow us to preserve endangered species where we simply don't have enough individuals to be sure that they're going to be producing offspring, or these individuals are spread out so far and few that getting them to mate is going to be an issue, or if they're just not behaviorally agreeing with the concept of getting more offspring being produced. So this might be one way to preserve endangered species. And it has been even proposed that we could, with this technique, potentially bring back extinct species, such as in Spain, they do have a mountain goat species that has become extinct. But by theory, if we do have the nucleus of a cell from that mountain goat species, and we're able to take only that nucleus and implant it into a blank cell, we could potentially make a clone of the material of the encoded into that DNA of the mountain goat, and in this way bring back a species. There has been some movies made about this idea. I think that most recently a remake of Jurassic Park and so on. So I'll let you on your own think about, are there ethical issues with this? Is this, how do you feel about it? But it's definitely something that technique is becoming more possible. Just to kind of wrap up the loop, I mentioned that few of my friends worked with the Dolly, and last I heard, I left Scotland quite a while ago. I believe it was in 2016, and what I heard at that time was that Dolly had 4 identical clones to her that were still at that time alive and doing fine, living healthy lives. So that's the story of Dolly the Sheep, and kind of giving a little bit, hopefully, relevance to what we have discussed in terms of techniques. What I would like to remind you is that these review sessions that we do together on Mondays and sometimes on Wednesdays, these are really aimed to give you the snapshot of the key concepts of the chapter material. For the full story with the full lecture videos, I refer you back to the learning modules that I have made available for you on the Blackboard here. And on those, I explain, for example, the process of producing Dolly, the nuclear transfer process, in a much greater detail. And with that, that brings us to the end of today's session. What I would like to ask you to do, and if you just hold on for a second, would be for you to post your name to the chat once more. This allows me to take attendance for anyone who was around for the entire session, or in case if you joined late, you still can get an attendance or so on. If you have to leave early, I will take the attendance from also the beginning of the session. So it's a way to ensure that anyone who was present will get the attendance. And that completes everything that I had planned for today's session. Like I said, I realized that this week's chapter and next week's chapter are bundled together. So I'm not expecting you to complete that quiz until when the quiz for next week would be due. So I'm going to be changing that due date if it's not already changed. I can't remember, and making sure that you have time to get also a review session of the next chapter. So we will not have a session on this coming Wednesday. We're probably all going to be looking forward to many other things and preparing for the Thanksgiving. So no session on the 17th, but we will see again on the 22nd, which is a week from now. And then we'll discuss about the chapter 15 and kind of expand on this journey from genes to proteins that we've already discussed. The quiz for these 2 chapters will be due on 23rd, so a day after our session next week. But again, if you do want to work ahead, you can do that, and you can study the material from the learning module and get that taken care of. And again, next week we will not have a session on the Wednesday. So that completes everything I had planned for today. If you have no questions, you're free to go, and I appreciate you having joined me. If you do have any questions, you can stay behind, and I will make sure to answer anything that you might have. Thank you for good night wishes. Thank you. Have a good night. I appreciate you. Yes, we have a question. Is that something you would like to ask in private, or are you happy if I still keep recording?
It's okay if you keep recording. I just wanted to know, why is Dolly the sheep named Dolly?
Dolly. Why is Dolly the sheep named Dolly? So Dolly was cloned from breast tissue of another adult sheep. And when these researchers were thinking of the name, when the cloning of Dolly was success and those cells started dividing, they were thinking of what would we call this individual? They thought about breast tissue and what would be an impressive example of that, and they went with Dolly Parton. So that's the story behind the name. I know it's a little bit of a dumb story, but that is a true story. So that's the answer to that. Did that help?
Yes, it did. Thank you.
Okay, thank you. It's just as simple as that, the question and the quiz. If you answer that, you're done. Any other questions? I'm here around if you have any questions. Yes. Would you like me to stop recording? Do you have a personal question, or are you happy to discuss them in a class? Yesenia?
Sorry, I had a personal question.
Okay, what I'm gonna do, I'm gonna stop recording, and we'll just check if anyone else has any questions. If they don't, then I'm just gonna close the classroom and just stay here behind with you so we can address that. But just before I'm gonna address Yesenia's question, did anyone else who's still present have any other questions? Anyone else than Yesenia who has questions? No, I don't see so. So what I'm gonna do now, I'm just gonna stop recording. And what I'm gonna do, I'm gonna clear the room. So Ryan, I'm not wanting to speak to you, but I'm understanding that you don't have any questions. So I'm just simply gonna move you out of the room so I can address the personal question that Yesenia had. And now it should be just us two here. So if there's anything I can help with just let me know.
Yeah, on the last conversation, our private conversation that we had the last session, I
Yes.
noticed because I was watching the lecture video again, and our whole private conversation is on the lecture video.
Ooh, is that, did you watch it from Blackboard or from YouTube?
Blackboard.
Okay, I will make sure to remove that from the Blackboard. I didn't know the students had access to that. So let me make sure that I remove that.
Okay, yeah, because I was just watching the lecture video, and I was like, our whole conversation was on there.
Well, I'm so sorry about that. I will make sure that I'll just probably remove the entire recording. I was not aware that you guys can get to that, so I'll make sure to get that taken care of right away.
Okay. Yeah, because I
Thank
usually
you.
go to the Collaborate, right?
Okay.
And I watch the lectures videos from there.
Okay, that makes sense. Yeah, and didn't have that opportunity before, so that's something that I'm still figuring out. So thank you for letting me know, and I apologize for that.
Oh no, you're fine. I left a message and I was like, well, I don't know if he's busy or not.
I
And you
might have
didn't get
missed
back to
that
me,
message.
so
Yeah, but thank you for bringing that to my attention, and I really do sincerely apologize for that, and I'll make sure that that gets blocked from there or made invisible for anyone else.
okay, all right, thank you. I just wanted you to be aware, and I was like, oh God.
Oh, that's super important. I don't want that to happen, and that helps me also with other classes and other students. So thank you for letting me know.
Oh, you're welcome. All
Have a
right,
good
thank
night.
you.
Bye.
Have a good night. Bye-bye.